Hello students, today let us learn about control of gene expression in prokaryotes. Let us start with the introduction. Gene regulation is the control of gene expression by various mechanisms that control the type and level of gene expression or the production of RNA or protein from the gene. Control of gene expression can occur at the level of transcription to RNA processing or at the level of post-translational modification of a protein. Prokaryotes also have regulatory mechanisms that control gene expression or whether a particular gene will be transcribed into messenger RNA or mRNA or not. Gene regulation in prokaryotes commonly involves the role of regulatory molecules that bind to DNA near the gene and help RNA polymerase to transcribe or block the transcription. In prokaryotes, the structural genes encoding the enzymes with related functions or those that catalyze the different steps in a single biochemical pathway are usually encoded together within the chromosome as a single unit or block called an operon and are transcribed together from one common promoter as a polycistronic messenger RNA. The expression of the structural genes of an operon are controlled simultaneously by regulatory molecules as these related enzymes are either needed all or not needed at the same time. Let us now see the anatomy of an operon. An operon includes the DNA sequences encoding the structural genes or regions coding for related polypeptides along with other regulatory regions that influence the transcription of the genes. In addition to the structural genes, an operon also includes the promoter and other regulatory sequences that regulate the expression of the genes. The promoter is the region where the RNA polymerase binds and is present upstream of the genes in an operon. The binding of RNA polymerase to the promoter is essential for transcription to occur, which will lead to the production of messenger RNAs. The regulatory sequences in the operon are DNA sequences that serve as binding sites for regulatory proteins, and the binding of these proteins to the operon by either turning it up or increasing or down or decreasing the expression of the gene. The regulatory proteins are encoded by genes called regulatory genes in the prokaryotic genome and are commonly of two types: repressors and activators. Repressors are those types of regulatory proteins that bind to regulatory DNA sequences called operators. The binding of repressors to operator reduces transcription or turns down the expression of the operon by blocking RNA polymerase from moving forward on the DNA. On the other hand, activators are regulatory proteins that, when bound to the regulatory DNA sequence, increases transcription or turns off the operon by helping RNA polymerase in binding to the promoter. The regulatory proteins can themselves be also turned on or off by the binding of specific small molecules. When these small molecules bind to the regulatory proteins, it leads to change in the shape of the regulatory protein that alters the ability of the regulatory protein to bind the regulatory DNA sequence. For example, an activator may become active or able to bind the regulatory DNA sequence only when a certain small molecule is attached to it. Let us now see the types of operons. Based on their level of expression, operons may be grouped into two main types, inducible or repressible operons. Inducible operons are the operons that usually remain turned off but can be turned on by a small molecule called inducer. For example, the LAC operon of E. coli that encodes the enzymes for the metabolism of the sugar lactose is induced by allolactose, the substrate of the pathway. While the repressible operons are the operons that usually remain turned on but can be turned off by a molecule called co-repressor. For example, the trip operon of E. coli encoding the enzymes for the synthesis of the amino acid tryptophan is repressed by tryptophan, the product of the pathway. In addition to the genes controlled by the two types of operons, some operons in prokaryotes containing the housekeeping genes are constitutively expressed or constantly active. The promoters and other regulatory DNA sequences of the housekeeping genes ensure constant expression of these genes since their products are needed by the cell all the time to maintain essential functions like DNA replication, repair and expression as well as enzymes involved in core metabolism. Let us now study about the LAC operon, which is an inducible operon. The LAC operon of E. coli was discovered by Francois Jacob between 1922-2013 and Jacques Monod, and they were awarded the Nobel Prize in Physiology or Medicine in 1965 for their discovery of the operons. The LAC operon contains genes and regulatory DNA sequences that function in the metabolism of the sugar lactose. The structure of the LAC operon contains a cap binding site, promoter, operator and three structural genes. The three structural genes are LAC-Z, LAC-Y and LAC-A. The gene LAC-Z encodes beta-galactosidase, which is an enzyme that cleaves the disaccharide lactose into glucose and galactose. The LAC-Y gene encodes beta-galactoside permease, which is a transmembrane symporter that pumps beta-galactosides including lactose into the cell using a proton gradient in the same direction. And LAC-A gene encodes beta-galactoside transacetylase, which is an enzyme that transfers an acetyl group from acetyl coenzyme A to beta-galactosides. The promoter is the site where RNA polymerase or the enzyme that synthesizes RNA binds during transcription. The operator of the LAC operon is a negative regulatory site which overlaps with the promoter region, and when a repressor protein binds to it, it blocks the binding of RNA polymerase to the promoter, inhibiting transcription. The cap binding site, on the other hand, is a positive regulatory site where a catabolite activator protein or cap in short binds commonly in association with a CAMP molecule, and the binding helps RNA polymerase to bind to the promoter, promoting transcription. LAC repressor. The repressive protein of the LAC operon is a small molecule of allolactose, which is an isomer of lactose. When lactose is not available, the repressor binds tightly to the operator region and prevents RNA polymerase to start transcription. However, when lactose is available, some of the lactose molecules are converted to allolactose, and the binding of allolactose to the LAC repressor changes the shape of the repressor, preventing its binding to the DNA. The LAC inducer. The allolactose molecule that triggers expression of the LAC operon by blocking the binding of repressor protein of the LAC operon acts as the inducer, since it can turn on or induce the operon, which is usually turned off or repressed. Catabolite activator protein or CAP. Catabolite activator protein or CAP helps RNA polymerase in binding to the promoter by binding to a region just before the LAC operon promoter and increases the level of transcription by acting as a glucose sensor. When the glucose levels are low, CAMP is produced inside the cell, and the CAMP binds to CAP, allowing it to bind to the DNA, leading to activation of transcription by RNA polymerase. However, when glucose levels are high, no CAMP is produced inside the cell and CAP can no longer bind the DNA, resulting in low levels of transcription. Thus, the working of the LAC operon depends on the presence or absence of glucose and lactose, and the regulation of the operon under different conditions may be summarized as follows. When glucose is present and lactose is absent. In this condition, the LAC repressor remains bound to the operator, and the low level of CAMP resulting from high glucose level also make CAP inactive, and it prevents transcription by RNA polymerase. When both glucose and lactose are present. In this condition, transcription occurs at low level, since the inducer or allolactose binds to the LAC repressor, releasing it from the operator, but the activation by CAP is not there as CAMP level is low due to the presence of glucose. When both glucose and lactose are absent. In this condition, transcription of the LAC operon will not take place, since the LAC repressor will be bound to the operator due to the absence of allolactose, although CAP is active due to the presence of CAMP resulting from the high glucose level. When glucose is absent and lactose is present. In such a condition, transcription of the LAC operon occurs at high levels, since the presence of the inducer or allolactose releases the LAC repressor from the operator, and CAP is also active due to high CAMP level resulting from the presence of glucose. Let us study about the trip operon, which is a repressible operon. The trip operon in E. coli was the first repressible operon to be discovered by Jacques Monod and colleagues in 1953. The trip operon contains genes for enzymes that catalyze the synthesis of the amino acid tryptophan. The operon is regulated in such a way that the operon is repressed when tryptophan is present in the environment, and the amino acid tryptophan acts as the repressor of the operon. When tryptophan is not present in the cell, the repressor protein cannot bind to the operator and operon is turned on, leading to tryptophan synthesis. However, when tryptophan accumulates in the cell, tryptophan molecules binds to the trip repressor molecule, changing its shape and help the repressor in binding to the trip operator, turning off the trip operon and tryptophan synthesis stops. Next, let us learn about the structure of the trip operon. The trip operon includes 5 structural genes that encode enzymes catalyzing tryptophan biosynthesis, along with a promoter or RNA polymerase binding site and an operator or binding site for a repressor protein. The 5 structural genes of the trip operon are TRPE, TRPD, TRPC, TRPB and TRPA, which code for the enzyme tryptophan synthetase. These genes are transcribed as a single messenger RNA containing the coding sequences of all the 5 genes, which are translated separately, and the proteins serve as the subunits of the enzyme tryptophan synthase. The trip operon contains gene called TRPR, coding for the regulatory protein repressor protein that binds to the operator in the presence of tryptophan and blocks transcription. The working of the trip operon under different conditions may be discussed under the following heads. High tryptophan level is present in the cell. When the level of tryptophan is high in the cell, tryptophan binds to the trip repressor protein and causes changes in its shape, converting to the active form, which allows it to bind to the DNA in the operator region. The binding of the trip repressor along with the bound tryptophan to the operator blocks the binding of RNA polymerase to the promoter and switches the operon to the off position, preventing unnecessary transcription of the biosynthetic enzymes. Thus, in the trip operon, the trip repressor acts as both a sensor and a switch that senses the level of tryptophan in the cell and switches it off when the level of tryptophan is high in the cell. The tryptophan molecule, which changes the repressor protein from its inactive state to its active state, is termed as a co-repressor. Low tryptophan level in the cell. On the other hand, when there is little or low level of tryptophan in the cell, tryptophan is not available to bind to and activate the trip repressor. The inactive trip repressor cannot bind to the DNA to block transcription, and therefore the trip operon remains switched on to transcribe enzymes for the biosynthesis of tryptophan. Next, trip operon regulation by attenuation. Unlike the LAC operon, the trip operon also contains a leader peptide and an attenuator sequence, which allows for graded regulation called attenuation. Attenuation is a regulatory mechanism that reduces or turns off the trip operon when levels of tryptophan are high in the cell. However, unlike the regulation by the trip repressor, which blocks transcription initiation by blocking the binding of RNA polymerase, attenuation prevents completion of transcription by causing RNA polymerase to stop prematurely so that it does not synthesize any tryptophan biosynthesis enzymes. Let us now see the conclusion. Thus, gene expression in bacteria is regulated by operational units called operons, which function by switching on or off the set of genes required for a metabolic pathway. Some operons like the LAC operon of E. coli that usually remain turned off can be turned on by small molecules called inducers, while some operons like the TRIP operon of E. coli usually remain turned on and can be turned off by a molecule called co-repressors. These two types of operons are called the repressible and inducible type of operons respectively, and they represent two main systems of control of gene regulation in prokaryotes.